Okay, Dr. Chandy, whenever you're ready.
You're muted.
Yes.
There you go.
Thank you all for joining on a very cold, wintry day today. We're here with the COVID-19 task force, and our goal today is several-fold, kind of update what's going on with the pandemic and the variants. The second goal is to talk about what the vaccines are and what the current data are for vaccine safety, efficacy, and some of the questions. And the third part of this would be to try to get some of our audience out here to become what we call trusted messengers, so they can go out and reach out to their communities, their ethnic communities, their area, their churches, and their local groups, so you all become experts in being able to communicate what we think is a very good vaccine. And we'll have, there are more vaccines coming out, and that will have to be adopted as we get more vaccines. So the first thing I wanted to lead into, where you see all the leads tonight on news, which are about the vaccine variants. And Dr. Johnson and Dr. Lynn are going to help me with this. So the next slide that you're looking at is the slide up there, Ruby?
Yes.
Okay, great. Is the South Africa, let me just minimize this screen. Excuse me for 1 second. Is the South African variants that are out there. There's several variants out there. Today you saw the news from South Carolina where there's the South African variant that was detected. The CDC doesn't want us to use the terminology of where the variants are from because they don't want to be pejorative towards the countries. The variant, the B117, is the one that where we broke away from as a country 200 odd years ago, which will go nameless for now. The other variant is the South African B1351, and now we have a Brazilian variant also out there. Why are these also important? It has to do with viral pressure, and it has to do with how much more easily they're transmitted. The big question out there is how efficacious the vaccines are that we currently have. And Dr. Johnson, could you comment a little bit about this? Because Dr. Johnson is a professor of pediatric infectious diseases and clearly knows a lot more about vaccine than I do, and he's done this for a lot longer than I have with vaccines.
Dr. Chandy is always very kind with his descriptors. Thank you very much. I think one of the important things has already been said, which is that it's not important where the variant comes from. What's important is that there is a variant, and variants are a natural part of evolution. We have to recognize that billions upon billions of viruses are being produced daily within even almost single individuals. And that creates an amazing evolutionary machine. And that evolutionary machine responds to immunologic pressure. And so because of the immunologic pressure, that means then that we see the generation of different variants appearing. If they didn't appear in one country, they would just appear in another. Remember that the original variant of the virus that came to the US was very quickly replaced by another variant, which was even more infectious, and now has been replaced by still yet another variant that has increased its infectivity. So we're going to see this variation occur over and over again. The goal of vaccination is to reduce the total burden of virus. In fact, you can really think about the driving force behind variants as being a function of just how much virus is circulating. The lower the amount of virus circulating, the better off we are. And this is the reason why vaccination is so vital. But it is, to a certain extent, a race against time. We know from other coronaviruses that eventually there are escape variants that come to infect somebody who's had a coronavirus before. So this was to be expected just based on that. We were hoping that we will be able to cover it with the vaccine, and so far our luck is holding in that every variant that's been identified still shows evidence of control by the vaccine's immune response. You may have read that the variant that have been described, at least one of them, showed that if you used convalescent sera from people who were infected very early in the epidemic, that it did not show effective suppression of the virus. But so far, with the current use of the two vaccines that we're using in the US, that they are suppressed by it. And so hopefully that will continue to occur. So time will tell.
Time will tell. So this is the big news because currently we have about 25 million people vaccinated, a little over that today. We also have about, some estimates are as high as about a quarter of our population now that has achieved herd immunity with a cost of over 400,000 lives, 430,000 lives thus far. And the next phase of this is we're kind of slowing down this last few weeks with the outbreak, with the surge that we've had, but the numbers, as well as the CDC believes, that come March and April, we'll have another surge one more time. And these mutations will become the B117 mutation they expect to be the predominant strain. So can we go to the next slide? So Dr. Johnson is going to talk about the mRNA vaccines and how they work.
Great, thank you. As we know, the two versions of the vaccines that have been approved for utilization through the emergency use authorization from the CDC, the FDA, has resulted in the availability of mRNA vaccines. So I just want to give you a very brief overview of an mRNA vaccine. And the good news is that mRNA vaccines have been in use and development for the last 20 years. Although this is the first vaccine that's had broad utilization, the mRNA format, in fact, the current type of mRNA format has been being investigated for about 10 years. And the news is really good because it appears to be a very safe and effective way of generating and giving people vaccine. So what happens with it? Originally, when mRNA vaccines were created, you were giving naked mRNA. You were just injecting mRNA. And that generated a very intense immune response because the naked mRNA was very immunogenic. But it also meant that it led to a higher rate of side effects than what we're seeing with the current mRNA vaccines. In fact, it created more severe side effects. And very little of the mRNA was able to get into the cell because so much of it was being destroyed in the bloodstream and in the tissue before it was being taken up by the host cell. So someone got the clever idea of putting a lipid membrane around the mRNA. And the value of that was first that it essentially hid it from the body's immune response, but it also meant you could use a much smaller amount of mRNA. And that reduced the severity of side effects. So what now is being used is a lipid-encoded nanoparticle that has mRNA inside of it. It's able to get to the host cell, be taken up by the host cell, as you can see in this cartoon drawing. And then what happens is that the endosome and the lipid membrane is broken down, and that leads to release of the mRNA. All of this is going on in the cytosol. None of this mRNA gets into the host nucleus, and therefore it does not have any impact on the host DNA. But it does what mRNA does, which is encode for protein synthesis. And that protein synthesis then leads to either secretion or a transmembrane protein that is immunogenic. Next slide. And I want you to think about the mRNA, the coating around the vaccine. It's kind of like an M&M. It's the same idea. That the M&M protects the chocolate. Well, here we are. We're just protecting the mRNA, so it doesn't come off in your hand in the case of chocolate. It means that the mRNA gets to the host cell. Next slide. So you inject it into the muscle. It's taken up by muscle cells. It's also presented to antigen-presenting cells. Next slide. And this leads to then a broad-based immune response. And that's the great news about this vaccine, is that it doesn't just generate antibody by interacting with B cells. It also encodes, leads to a broad-based T cell response. And both of those are critical to the success of this vaccine. And that broad-based response is what's protecting us. Next slide. So I'm going to turn things back over to my partner here, Dr. Chundy, who will take us forward.
Dr. Johnson, one question about the 2 doses and the can you go back to the slide before? Yeah, so the CD8 response, there's been some conversation about 1 dose and delaying the doses. Can you comment on that and why 2 doses are important for this vaccine?
Yeah, so one of the things that we know from many different vaccines is that you need a priming step and then a boosting step. And the boosting step, of course, that takes advantage of the broad-based immune response in order to strengthen the level of antibody formation that ultimately comes from the vaccine. So we know from some of the data that's been generated that maybe about 50% of people give an adequate response to the first dose of the vaccine, and that they need re-exposure to the antigen in order to strengthen the response and lead to a stronger antibody response. We're hoping with the J&J, the Johnson & Johnson vaccine, no relationship to me, that the Johnson & Johnson vaccine can succeed with a single dose. But you got to remember, it's a different vaccine. That is not an mRNA vaccine. That is an attenuated adenoviral vector vaccine. And the beauty of that is that it leads to some replication of virus. Eventually, it's gone, but replication of virus then leads to an ongoing immune response. And it seems that the immune response actually strengthens over time with the Johnson & Johnson vaccine. So that if you look at the data 2 weeks out versus 4 to 6 weeks out, you actually see a heightening of the immune response. That's the idea of giving a second dose of the mRNA vaccine.
Thank you for that explanation, because this is a critical kind of thought process that you do absolutely need the second dose. It may be delayed for a little while, but not, for months, maybe a week or 2.
Right, and the big reason why some countries are saying they want to delay is because if you do some statistical analysis, you can show that over the first few weeks of rollout, you're vaccinating and actually making immune a larger percentage of the population than if you're holding back that second dose in order to ensure its presence. So by 6 to 8 weeks, though, you really want to be giving that second dose anyways, because then there really is very little difference between using it one way versus the other. So all those places that are using 1 dose right now, they're going to use 2 doses. They're just delaying it in order to cover a little bit larger percentage of the population.
So just to clarify, the J&J vaccine is a spike glycoprotein. It itself doesn't contain a whole coronavirus.
Correct. It's definitely not a live coronavirus vaccine. Thank you for pointing that out, just to make sure that there's no confusion. None of the vaccines that we're talking about for use right now in the US are attenuated coronavirus vaccines, or coronavirus vaccines for that matter. And so
The
please
adenovirus
don't confuse it.
Right.
That's right. The adenovirus is what's attenuated. Or it's a mammalian adenovirus, but not a human adenovirus. So for example, the Oxford vaccine is a chimpanzee adenovirus.
Thank you. Next slide. Keep going. Next slide. So one of the goals for this talk today is to try to get the people that are participating to become fairly well-versed in vaccines so they can go talk to the lay public and become a trusted messenger. And the reason why this is kind of critical as a medical society and as physicians, as well as healthcare providers, is to be able to go out to our school boards, our churches, our local folks out there and communicate with them about what vaccines are and how important they are for to basically get rid of this horrible 10 months that we've been living through. And I think we want to understand mRNA vaccines and be comfortable communicating with your patients and constituents. Become an expert and be armed with knowledge to explain the myths. There's a lot of myths going on and fallacies to convert the vaccine hesitant to vaccine acceptance. In fact, the New York Times had an article about how if you've seen, the Chinese government has been out there trying to blunt the belief that these started in China, and they're actually helping some of the anti-vaxxer sites. I quite don't understand all of this, but that's what's happening. And so we want to be truthful. We want to have knowledge on our side. Next slide. So one of the things that you saw from, I'm gonna have to unplug my little computer here. It keeps going on and off. There we go. One of our, now I can't see the slides. I'm sorry, Natalie. Let me push my little thing here. Okay, so one of the questions that's always comes up is, I have natural infection, why do we need the vaccines? And natural outcomes of COVID are pretty serious. We've seen the 400,000 deaths, which I can't believe I'm saying, even when I say it. The important thing is even young, healthy people can get very ill. These are two patients that I've taken care of. One was a 42-year-old gentleman who came in with 8 days of fever and cough, shortness of breath. He was otherwise healthy except for fairly well-controlled diabetes. And within a week and a half, he went from nasal cannula to high-flow nasal cannula to BiPAP, intubation, eventually got extracorporeal membrane oxygenation, and eventual lung transplantation. The other one was a young 19-year-old woman who presented with headaches, fevers, chills, and actually presented with a shock-like syndrome. She looked like she had toxic shock syndrome and had liver function abnormalities. And for all the world, she required pressors and was found to be actually COVID positive for antibodies. So she had a COVID infection a while back, and what she ended up having was this multi-systemic inflammatory syndrome in children. And she wasn't really a child, she was a young adult, or an adult in my perspective. And so these are the sorts of things that natural infections do, and which we should avoid by immunizations. And the interesting thing with the immunizations are actually, the current vaccines we have actually give you a better immunity than natural infections. Not everyone who gets infected develops antibodies or develops protective antibodies. If you do get antibodies to the spike glycoprotein, they seem to be protective from natural immunity. But a lot of the people who've passed away never really develop good spike glycoprotein antibodies. They get nucleocapsid antibodies, which are not protective. Go to the next slide. And why is this such a big thing? Because you're going to have a lot of the public asking you, well, because they don't remember the good old days when we had iron lung machines for all the people who've had polio. There's a much higher incidence of complications of natural infections compared to vaccines. That's why we have vaccines. And that's why when Jenner started with the cowpox vaccines to prevent smallpox. In fact, one of the big sayings used to be, "I pox on your family," in the 18th century. It was like cursing out a family because the smallpox was such a dreaded illness. And vaccines are always safer than herd immunity. We've had I think Senator Rand Paul talking about how natural herd immunity is really strong immunity. You had COVID parties for people trying to get infected with COVID, which is really stupid and dangerous. And so the vaccines are designed to be much safer than the infection. And the vaccines paradigms are different than what we do with treatments. Vaccines have to be much safer because you're giving it to a natural, healthy a healthy population. And vaccines undergo the most rigorous sort of trials with tens of thousands of participants, and the degree of safety has to be extremely high. And to gain approval, all vaccines undergo these trials, and only 1 in 10 candidates make it to the biologics license applications. We've already had the Merck vaccine, which bowed out this week. We had the GSK vaccine bow out, and there was one other that also did not make it through the cut thus far. Next slide. So Dr. Johnson touched on this. So the blue is the mRNA. Basically, it does not get into your DNA. I can't emphasize this enough. It goes into your cytosol, and your ribosome starts making the protein that the mRNA encodes for, and in which case it's the part of the spike glycoprotein, which is the immunological aspect of it, and then your body starts making antibodies to it, as well as stimulating your T cells to remember and to bring back memory for this virus. And that memory is important because it gives you long-lived immunity. That's why that second injection for these mRNA vaccines is so important. The mRNA platform was originally designed for cancer as well as for HIV. The HIV vaccine did not work out so well. It was also an adenoviral vaccine, and we're still trying to figure out how to fight HIV, and there's lots of difficulty with that. And the mRNA cannot be integrated into your DNA. It does not change your DNA in any manner. Next slide. So why did these vaccines come up so quickly? The use of this terminology of the warp speed was not the greatest, because it kind of lent itself to saying we rushed through things. It was not rushed. It was actually concurrently developed, and the government guaranteed that the companies would not have to face the risk of financial failure. And so companies went out there and started developing these vaccines quickly. The Zika vaccine, remember Zika virus? The Zika was this virus that caused all sorts of birth abnormalities. So Zika virus was out there, and there was 2 vaccines that were developed, interestingly, by the same 2 companies now, Moderna and the German company, which Pfizer bought, and the BioNTech. And they developed these vaccines because they thought Zika was going to be the massive plague that we have currently. But interestingly, Zika just kind of disappeared, and the reason why Zika disappeared was because dengue basically took over the mosquito vector that Zika uses, the Aedes aegypti mosquito, and didn't give Zika an ability to go between humans. But the platform that was developed is exactly the same. And this platform helps. It can be basically a plug and play sort of thing, like Legos. And when the Chinese government, the researchers put out the whole gene sequence of the SARS-CoV-1, the NIH came out within a week, I think, with the antigenic portion for people to develop the vaccine. And within 6 weeks, they've actually developed the vaccines that we have today. And then the testing went on with the NIH, which already had a well-developed, vast network of trials. So all of this happened concurrently. And when the vaccines showed some efficacy, the federal government took the risk and said, okay, we're going to Moderna, here we go. We're going to give you money to go ahead and produce the vaccine. And in the Pfizer case, Pfizer did take the money and said, we're going to produce it ourselves. And so the vaccine production took place at the same time the trials were going on. Thus, we were able to enroll people quickly and then get the vaccines out. Next slide. So the safety was demonstrated. The primary endpoint was the incidence of COVID-19 infection. Secondary endpoint was hospitalizations or death, and these endpoints were met. In the Pfizer vaccine, over 43,000 participants. In the Moderna vaccine, 30,000 participants. There were no major serious adverse effects during the trial, which is astounding on both the Moderna and the Pfizer side. There were these strange, now they're calling anything anaphylaxis, but it's really anaphylactoid type of reactions. At that time, it occurred in Britain. Now we've seen a fair amount of this, but we've given 25 million doses of vaccine. And there is one reported case of someone having thrombocytopenia in Florida who passed away, physician, and there are these reactions that you get after the vaccines with rashes, which are well described. The VAERS database is chock full of people having local reactions, and then these people who have anaphylaxis to other vaccines having significant reactions. And so there's now people have to actually wait for 30 minutes if you have these severe allergic reactions. And that's why you should try to tell your patients to stay there for a minimum of 15 minutes if they have no reactions, and 30 minutes if they have a history of severe reactions. And for the next slide. So the efficacy of this vaccine is over 95%, which was astounding. I mean, the WHO said if you went over 50%, we'd be happy. We're at 95%. So it really gives a high degree of protection. As of 2 weeks ago, it was 16 million doses. Now it's about 25 million doses. We're doing about 1 million doses a day in the country. There is anaphylactoid reactions are occurring for both vaccines. There has been no Guillain-Barré to date, no transverse myelitis to date. There are these small fiber neuropathies. There are these other things that are described with change of taste in the mouth. There are reactions at the local site where you get local injection site pain. I mean, I have had both doses of the vaccine. The first time I took it, all I had was really arm pain and a little bit of chills and rigors. The second dose was a doozy. The second dose, about 24 hours after the second dose, you start having chills and headaches, body aches, which just basically says my immune system has woken up and it's recognized this virus. And in my case, I had COVID in April, May. And so I think I had a little bit stronger reaction because my immune system was already primed. And then the other strange reaction that we're seeing is this delayed response after 7 days. And Dr. Catherine Mullane said that this is due to a subcutaneous injection. Dr. Offit believes it's actually not CD4 mediated, but cytokine mediated. Today, I was reading it from Mass General on the dermatology department that this just should not preclude you from getting a second dose of vaccine. So that should not stop you from getting a second dose of vaccine. Next slide. So there are two different vaccines. You cannot mix these two vaccines. You can't take these vaccines and say, okay, one day you took Pfizer, the second time you took Moderna. The Pfizer vaccines is a bit more difficult to handle. It's very cold, -70, and so you can basically hurt yourself handling the vaccine if you don't have the correct type of gloves. It's stable for 6 months under ultra cold storage. And once you thaw it, you're done. You have to use it within, and you reconstitute it, you have to use it within 6 hours. It's better for hospitals and urban centers with access to very cold fridges. Undiluted, it's good for 5 days in your regular freezer in your office. Once it's diluted, you got to use it. So it's quite a difficult vaccine to handle. The Moderna vaccine is stable for 6 months at -25 to 15 degrees. And it cannot be refrozen once it's thawed. and it should be stored at -40. Better for private doctor's offices. If it's unpunctured, if you don't put the needle in, it's good for 30 days in the regular fridge and 12 hours at 8 to 25 degrees. And once it's punctured, it's good for 6 hours. So these are tough vaccines. I'm sorry, I made an error with the Pfizer vaccine. It must be used within 30 minutes after reconstituting and pulling it up. So there's a lot of logistics to this. And just imagine if you're running a vaccine center where you're trying to give 10,000 doses of vaccine in a day, how it's got to be run like clockwork to get this correctly. Next slide. So the comparisons of both vaccines, these are very safe vaccines and they're very effective. And the Pfizer vaccine, most of the reactions are local. You know, the fever occurs in about 7.5 to 31%. You get local pain about 71%. Swelling occurs, redness occurs. You're tired after the vaccine, and you do get myalgias and chills. There's this lymphadenitis that you feel especially after the second dose. It's more common in Moderna than Pfizer. The Bell's palsy was reported, but it's no higher than what would be expected from a background population. So these are very, very safe vaccines, and most of these last for 24 hours. There was a question about taking antipyretics, Tylenol preemptively. I don't advise that, but you can certainly take antipyretics if you have symptoms. Next slide. And the indications are a little bit different. Pfizer was tested at ages 16 and older, so it's approved for that. The dose is different. It's 30 micrograms. The efficacy we talked about again is over 95%, and it does cover both the UK variants and the South African variants. And these vaccine trials were excellent for another reason. They really did include the cornucopia of all the races we have in America. There was a good representation of African Americans, a good representation of Hispanic population as well as Asians. So it really did represent America. And protection, the protection starts about 10 days after the first dose. Even though the protection, they say, is up to 80% after a single dose, you really need a second dose because the durability is really important to get from the priming dose to get to the second dose. And it does protect you against severe disease and death. And the number of cases and the study population was small, but now we have 25 million doses given. It's very safe. The Moderna vaccine, again, it's a different vaccine. You can't mix them. 100 microgram dose, you give it on day 1 and day 29, not day 1 and day 21 like Pfizer. The efficacy is excellent. It's indicated for 18 and older, and it's included in the. It's a little less effective against the South African variant, and they're actually, both companies are reconstituting. They've actually are about to come out with booster doses that would cover both of these, but these variants are changing, and this is going to be an ongoing battle if the level of infections stay at this rate, because there's a tremendous amount of pressure for this virus to mutate and survive. Protection starts about 14 days after the first dose, and the durability of a protection is unknown, and onset of first symptom and the protection is about 80 to 90% by the time you get to 28 days. And it does protect you against severe disease and death. So the next slide. This is from the FDA approval process on December 10th for the Pfizer, and Moderna it was December 30th. You can see the efficacy of the Pfizer vaccine takes off a little earlier than the Moderna vaccine, but by 2 weeks after the second booster dose, the rates are about similar, about 95% vaccine efficacy. So you really should wait for 2 weeks after the second dose before you're very comfortable with your vaccine efficacy. Next slide. So what is this trusted messenger and why is it important? If you look at who is really vaccine hesitant, they're the groups that are most affected by COVID. It is the African American population. It is the Latinx population. The areas where people are running into line to get vaccinated are the people who are generally living in the suburbs, exurbs, and they're getting vaccinated at a high rate. And they're the people that are dying at an excessive rate are really hesitant to get vaccine. And this has a lot to do with what's been going on over generations of not trusting in the postmodern world, the government, the not trusting physicians who they're not connected with. And who do they trust? They trust the physicians who live in the areas that they're at. They trust the local clergy. And they trust the folks who are on the ground, the case managers, the social workers on the ground in those communities. So we want to get these folks to be the trusted messenger and to be the motivators for getting vaccinated. In some of the hospitals in predominantly minority areas, more than half of the healthcare workers do not want to be vaccinated. And that's a big problem. Even in the nursing homes, a lot of the minority communities, the workers do not want to be vaccinated. And so it's really important that we get these vaccine hesitant people to become vaccine acceptance people. And the context is really important for this. There was a Nobel Prize winner in economics who looked at peer pressure. Peer pressure is real and it works. And that's why all this social media works so well. And getting the word out, being the agents of change is really important. So everyone taking the vaccine is important, not just, the NBA has Kareem Abdul-Jabbar out there who's over 65 getting his vaccine. They have some of the older coaches out there getting the vaccine. And these are really important messengers to affect change. Next slide. So some of the questions that are asked is, if I had COVID-19, do I need the vaccine? We get asked this question all the time. Yes, because natural immunity wanes. Not everyone who gets infected develops immunity. And it has to do with how the older you are, the less likely you're going to develop effective immunity with natural infection. The sicker you are, if you get really, really ill and you're in the ICU with COVID, you may not develop the effective antibodies. Will the vaccine alter my DNA? Unequivocally, no. RNA does not enter the nucleus and does not alter your DNA. Will the vaccine make me sick with COVID? It does not make you sick with COVID. We've had now multiple cases of people who've gotten the first dose of COVID vaccine and then got exposed to someone with SARS-CoV-2 and became ill with COVID. And they basically had a minor infection and got better. And then the correct answer is to go ahead and give them the second dose of vaccine. But the vaccine itself does not make you PCR positive. It may make you antibody positive. The feeling after vaccination of body aches and feverishness is because your immune system is awake and it's causing your T cell activation, your antibody production, and you get cytokine release, so you don't feel great after the vaccine. Next slide. And then, will the vaccine turn my COVID test positive? And the answer is no, it does not turn the PCR test positive, the nasopharyngeal swab or the salivary test, but it may turn your antibody test positive depending on which antibody it checks for. There are antibody tests that basically check nuclear capsid antigen, so antibodies, so it wouldn't, some of those tests would not turn positive. And then if I'm vaccinated, why do I need to wear a mask? And this has to do with the difference between neutralizing antibodies and sterilizing antibodies. Clearly, the vaccines are very effective in preventing disease and death, but you can carry the virus in your nose, and you can secrete small quantities of virus and possibly transmit to somebody in your household or to your patients who are immunocompromised. So you do need to wear a mask until the level of COVID in the community comes down to a point where there is the R naught becomes less than 1, and you have low levels of actual virus circulating in the community. Next slide. So are there people who should not receive vaccine and mRNA vaccine? And the answer is yes, if you have a hypersensitivity to the components of the vaccine with this polyethylene glycol. So if you have immediate allergies to this polyethylene glycol, you should not receive the vaccine. is a history of anaphylaxis to any other vaccine or injectable therapies is not a contraindication to getting the mRNA vaccines. So those folks, like we talked about earlier, should be observed for 30 minutes in a controlled circumstance. If you receive plasma or you receive monoclonal antibody because you got sick with COVID before, you have to delay it for about 90 days so you don't block the uptake of vaccine. And the likelihood of reinfection in those 90 days is extremely low. And the newer antibody preparations may have longer half-life, so they're going to adjust the time for the revaccination based on which preparation you got. Next slide. The components of the vaccine are listed there for people to see. The magic potion on these two vaccines is actually the lipid molecule on top. These are nano pegylated particles, which are also causing most of the reactions. And the Pfizer differs because it contains a different stabilizer, and that you can look up. This is for folks to look at. The next slide. And then can people with impaired immune systems receive the vaccine? These are the people who really do benefit from the vaccine. And the most important thing is the risk of the mRNA vaccine is very low. The risk of getting COVID-19 and dying is significant. The increased risk of immune responses may be blunted if you're on high-dose steroids, if you're a transplant patient and you're on calcineurin inhibitors, if you're on all these other agents that we're talking about. Blood disorders and blood cancers and other cellular type of therapies, bone marrow transplant patients, solid organ transplant patients, HIV patients, they should all get vaccinated. Next slide. So the vaccines are safe, they're effective, and what we talked about, it's a complex cold chain, and especially for the Pfizer vaccine. You can see it's like a dance of how quickly you have to prepare it and get it out to folks. 2 doses of the vaccine, which makes it very much more complex, and they can't be interchanged. And you have about 300 million doses purchased. I think the Biden administration just put out an order for another 100 million doses. So you'll have, 300 million doses means 150 million people can get vaccinated. And the J&J vaccine, the Johnson & Johnson vaccine, is a single dose vaccine, and I think they're going to make up for the other doses. So you'll have enough vaccine by the end of the year. Next slide. What we don't know, how long the vaccine will protect you, and will the vaccine prevent transmission of infections? We don't really know the answer to that because the studies haven't been done, but you do have less viral load in people who get vaccinated, and most vaccine experts believe it will decrease the amount of transmission. Use in children, both vaccines are now, both Pfizer and BioNTech can now be used down to age of 16, and the studies are ongoing for younger children, but children generally don't get as sick. So right now the emphasis is to get adults vaccinated. Use in pregnancy and lactation. Natural infection is worse than risk associated with the vaccines. So Dr. Lin is going to talk about why pregnant women and lactating women should get the vaccine. And long-term side effects. Most vaccine reactions occur within 6 weeks. We have fairly good data that this is a very safe vaccine. Next slide. And the unknown knowns, we're gonna figure this out. Right now, it seems to be pretty safe. Now that we have a good month and odd with both of these vaccines, the incidence of Guillain-Barré we have not seen it reported yet, so it seems to be a very safe vaccine. Next slide. Dr. Lin, you're on. Dr. Lin is a professor emeritus of OBGYN and a chair and a former department chair of OBGYN at Rush. And Dr. Lin will talk about the ACOG practice advisory and pregnancy and lactating women.
Thank you very much, Dr. Chundy and Dr. Johnson, for this excellent presentation. I hope I will enlighten you a little bit about the reproductive issues. And I want to also thank the Chicago Medical Society for making this happen. May I have the next slide, please? My comments are really drawn from the American College of OBGYN and the Society for Maternal Fetal Medicine. Actually, they just released a joint recommendations regarding it. And I think, again, I just want to comment and emphasize the point that vaccines are critical as a public health strategy for prolonging life and removing the risk of very severe diseases. And I'm going to put this current vaccine into that group. I want to also emphasize the fact that, as was pointed out before, just because the vaccine appeared to be brought out quickly, as was pointed out, these types of vaccines were in existence for a long time. No shortcuts were used, and all the safety monitoring has been extensive. Now, when you're counseling patients about women who are interested in getting pregnant, women who are pregnant, or women who are lactating, again, I want to emphasize that the COVID vaccines do not and will not and cannot cause the infection. It is just simply a fascinating biological phenomenon where you incorporate a small sequence of RNA and you actually reprogram a cell to produce the protein that you're trying to make the immune response against. And it also that the vaccines have been extraordinarily safe, and the side effect profile has really been quite commendable. And it's continuing to be monitored as we're speaking. And for decades, we've had vaccines such as the flu vaccine, which we routinely give to pregnant women and lactating individuals. And that's a completely different type of vaccine that involves attenuated virus. So may I have the next transparency, please? So the recommendations in pregnancy are such. But the American College and the Society for MFM recommends that COVID vaccines should not be withheld from pregnant individuals who meet the criteria, and the criteria we're talking about is the 1A, 1B, 1C, the criteria that we talked about in terms of priority groups. At the same time, I don't think we need to coerce or to harass our patients who are somewhat ambivalent about this, but rather try to educate them a little bit. Now, we're at a disadvantage, and the public should be counseled and aware of the fact that when the vaccines were in clinical trials, pregnant women were not included. And therefore, we do not have the type of safety data I know that all of you would be extremely comfortable with. But also, there is no data to indicate that the vaccine should be contraindicated, and no safety signals from the DART studies from the Pfizer-BioNTech vaccine have arisen. But women need to make a free choice. And the decision should be in conjunction with counseling from their healthcare providers. And we should be free to offer these patients as much information as available. And I will direct you to the ACOG, the ACOG website where this information is readily available. May I have the next slide, please? Now, in your consultation with your patients, in the discussion, I'm going to urge you to emphasize a couple of points. One, and probably the most important thing that I want to say tonight, is that I'm very comfortable with the safety profile and the biological reality of these vaccines, especially in comparison to the natural disease. Because we do know that the COVID-19 infection in pregnancy can be complicated. And it does lead to significant morbidity in this population. And therefore, that has to be weighed against the theoretical concerns that we have from the lack of safety data involving pregnant women. So I think we have to also look, when you're counseling the patients, at the level of activity of the virus in the community. And as we all know, there are different areas of even the city of Chicago and the Chicagoland area that are disproportionately affected by the infection, and also disproportionately associated with hospitalizations and death. So that has to enter into decision making. We know how effective these vaccines are. They're really kind of remarkable considering other vaccines which we readily utilize are not nearly as effective. And we also have to look at as I said, the biological reality that we're not exposing the patient to anything that would be embryopathic or would cause disease that would jeopardize the pregnancy. May I have the next transparency slide, please? We have similar line of reasoning and similar recommendations involving lactation. And again, these vaccines should be offered to lactating individuals similar to non-lactating individuals. And we have a similar issue. They were not routinely included in clinical trials, but I can tell you that registries are being created to try to identify those individuals that did get pregnant, those that did have babies who were vaccinated, and looking at the outcomes. But I would not recommend withholding a lactating individual who otherwise meets criteria, who would benefit from avoiding the native infection. And because again, I'm reemphasized that the theoretical concerns for lactating individuals who are vaccinated do not outweigh the potential benefits. And there's no need to avoid the initiation or to discontinue breastfeeding of patients who receive the vaccine. May I have the next slide, please? Now, a couple of points. I know there were some questions raised in terms of infertility and fertility. There are no data that I'm aware of, and I look at this all the time, where there has been any proof that fertility is compromised. And I'm going to classify some of these rumors in the mythology category right now. And unfortunately, the internet and discussions are filled with misinformation, some of which is just individuals who are concerned voicing their opinion. But also there is, I think, some malicious anti-vaccination material that's getting out into the public's hands. And the vaccination is strongly encouraged for non-pregnant individuals using the same prioritization. And women who are actively trying to become pregnant or are contemplating pregnancy in the near future, and they meet these criteria, should be offered the vaccine. It's not necessary to delay pregnancy after completing both doses of the COVID vaccine. This is not like rubella, where we actively surveil our patients to determine whether they have rubella immunity and make an attempt then to avoid pregnancy during the 3 months following the vaccination, because that's an attenuated virus. Again, this is not what these vaccines are currently about. If an individual does become pregnant after the first dose, again, the second doses should be administered as indicated with the appropriate time interval. Now, with the government, the CDC has requested and have created what they call the VSafe website. You can get more information about this, VSafe program on the CDC website. And there's even an app that can be downloaded, because they want to get reports from the individuals who in fact were vaccinated to continue to increase our knowledge about these very important and at this moment, because of the concerns about persistence of this infection at this rate that we're experiencing, I think critically important strategies from a public health perspective. I believe that's my last slide, and I'll turn things back over to, I believe it's Dr. Chundi's turn again. Oh, by the way, I just want to comment. We did put the website here for you to register if you want to become one of our trusted messengers. The Medical Society is trying to cultivate a group of individuals who would be interested in reaching out to your constituency and your communities where you live and work, and who have established a trusted patient relationship with others. And I think that if you're interested, we're going to be more than glad to provide you with educational material that you can use in your interactions. Next slide. Are we done? Vishnu, do you have more FAQs?
No, there were a couple of questions that were out there that I think we should respond to. One was the timing of other vaccines. So if you had a pneumococcal vaccine or if you had an influenza vaccine, Dr. Johnson, if you can take that, when can you get your COVID vaccine?
So the recommendation that the CDC has put forward is that you wait at least 2 weeks after immunization, and you wait at least 2 weeks before immunization. So in other words, if you got a pneumococcal vaccine or any other vaccine 10 days ago, then you're going to have to wait another 4 days before getting the vaccine. And if you got a COVID vaccine, then the recommendation is to wait at least 2 weeks after you received it before getting a vaccine. They also, in case this question comes up, there was also concern about whether or not you can do tuberculosis testing with an IGRA, interferon gamma test, so quantiferon TB gold. And the recommendation the CDC has put out is to wait at least 4 weeks after vaccination before doing a quantiferon test. Although interestingly enough, even though it works by the same mechanism, they did not say that you necessarily needed to wait that long with a skin test. But they did recommend with quantiferon to wait at least 4 weeks. The alternative would be to just draw the test at the same time as you give the first COVID vaccine. Not that there's any relationship between tuberculosis and COVID, nor any known interference between the COVID vaccine and quantiferon testing, but out of an abundance of caution, they're recommending this type of delay.
The second question was about fertility, both in women and men. There's a lot of, in fact, using sterilizing immunity was probably the wrong choice of words there. Because I'd just like Dr. Johnson or Dr. Lynn to comment on the fertility aspect of this, because there's a lot of stuff on the internet about this.
Dr. Lynn's really the expert there.
Well, I will speak for women particularly, and right now, as I say, I am not aware of any valid scientific association between diminished fertility and people who receive this vaccine. I'm not as comfortable commenting regarding males who may respond to antibodies, but again, there's nothing structurally in the spike protein antibodies that should theoretically be interfering with spermatogenesis or adequate sperm motility. But again, that's a question that I don't have any personal experience with other than what I've been studying at this point.
Dr. Lynn, there's another question that's in your domain, which is, are there still any concerns regarding the similarities between the COVID spike protein and the amniotic membrane?
Not that I'm aware of. And again, it's right now, I think the biggest issue is that patients are exposed to a infection which has led to prematurity, and potentially there's some suggestion that possibly even loss of pregnancies at an earlier gestational age.
And Dr. Butt, or Tariq Butt, is the president of the Chicago Medical Society, and he is online. So Dr. Butt, Natalie, if you can let him in so he could say a few words, because it's 7 o'clock. Is he there?
Thank you so much, Dr. Chandi, and thank you so much, Dr. Chandi, Dr. Johnson, Dr. Len. Excellent presentation, like always. We are so glad so many folks could join us, so many physicians. And our Trusted Messenger campaign is one of the leading campaigns that we have to really lead with you and with clergy and with everybody who can serve as Trusted Messenger. So thanks very much for joining, who have joined. I got a lot of messages that they have joined already, Trusted Messenger campaign. If you haven't already joined us, please join us. And I like to thank you again for joining us. Thank you, Dr. Chandi, Dr. Len, and Dr. Johnson.
Thank you. So our goal is we're going to work with the city of Chicago and get everyone connected with their local communities, have a slide set, have question and answer material for you, and have an active area where we can answer questions. So thank you all for joining. Have a good night and shovel some snow this weekend. So there you go.
Thank you.
Thank you.